Thank you, everyone, for attending the Molecular Information Systems Lab's Colloquium today. We've got 5 students from the lab. They're going to be talking about a range of projects from molecular computing, storage, as well as molecular sensing. So the format will be around 10 minutes of presentation time, and then a couple minutes of Q&A afterwards. After each talk. All right, and with that, I'll introduce the first speaker, Karina Imbergia, who is going to be talking about CRISPR-Cas9 for similarity search in DNA data storage systems.
Cool. So hey, everyone. I'm Karina. I'm a second-year PhD student working with Jeff Navala and the MISL group. And I'm going to talk about CRISPR-Cas9 as a tool for similarity search in DNA data storage systems. And before I begin, I just want to acknowledge all the people who have worked on this project. A huge amount of the work was done before I even joined the lab, and I'm super grateful to everyone who's been a part of it. So why do we talk about DNA in the context of data storage? So data production is estimated to reach, on a yearly level, 175 zettabytes by 2025. And global demand for data storage is already outpacing capacity. DNA has emerged as an attractive medium, one, because it's dense. So in this image here, that faint pink smear can hold about 10 terabytes of information. It has great longevity, which means it's super robust to degradation over time. And then the technology around it, so DNA sequencing and synthesis, has eternal relevance as motivations to improve it parallel those in the biomedical field. So the overview of DNA data storage, it starts with the binary representation of digital data, which is then partitioned into sequences, and then we add an address to it so that we can reconstruct those sequences later. We then apply an encoding scheme to convert the binary into DNA base pairs, and we can synthesize those strands as DNA. We then store them for when we want to access them later, so they can be stored for indefinite amount of time. And so when we want to decode it or selectively retrieve information from our database, we can apply different methods to either retrieve the whole data set or a subset of it, and then we sequence the DNA and apply algorithms to reconstruct the data itself. And so an important thing here is that, depending on how we want to access the data or operate on it, we can change the encoding architecture in step 2 to facilitate our goals. And so this selective retrieval step that we see at the bottom here, it really becomes an information processing step as we introduce parallel computation to our data set. And so if we think about what makes an efficient database, we do need a way to retrieve subsets of that information. And this is particularly the case with DNA data storage, because if we want to operate on a subset, if we want to compute on a subset, we don't want to have to sequence the whole thing and store that all on the computer. And so one way to do that is with random access. But what if we wanted to just retrieve a subset of information that's similar to a query? So similarity search, you can think about Google's reverse image search, for example. So let's say we have a database of images, and we want to extract all the images similar to a query. Our goal would be to design an architecture that will encode similar images in a way where if we introduce a query also encoded in that way, it can facilitate molecular interactions that will allow us to decode only the representations of the images that are similar to that query. So previous techniques were developed by people in this lab and used DNA hybridization, but it was thought that maybe Cas9 could be an effective alternative to that because it's a much easier protocol to perform, and it uses an isothermal reaction, which means that it's very energy efficient. So Cas9 is a programmable nuclease that's derived from a bacterial immune system. So it's a lot of kind of biology jargon. What does that actually mean? So we can break it down. So a nuclease is just an enzyme that cleaves nucleic acids or DNA. And then what we mean by programmable is that we can design a guide RNA that integrates with the Cas9 system to match a target region within the DNA that we want to cut. And so Cas9, our programmed Cas9, can be introduced to our DNA database, and it can sit and make a cut on the matching sequence depending on how we designed it. And it will cleave that sequence into 2 fragments. So the important thing about Cas9 and what we wanted to leverage for our similarity search architecture is that it won't just cut an exact match from the guide RNA query to the target. It'll also have a number of off-target activity and cut things that are sequences that kind of match or just similar to it. And so we wanted to leverage this. In other words, we wanted to encode a data set in a way that maps content similarity of what's in the database to similar sequences so that Cas9 can come in and target those. So the first step of that is to train a model that will do this encoding. And so what we did is we extracted feature vectors from the FC2 layer of an image classification model. And this produces these feature vectors, which are just semantic embeddings of images, and they act as a summary of the image. And so that we can then use the Euclidean distance between these feature vectors as our metric for similarity. And so the images are encoded into these sequences, and then we use a cleavage predictor model, which predicts Cas9 activation between pairs of sequences. And then it computes a loss, which is then backpropagated to train the encoder to maximize the activation of similar sequence pairs and then minimize activation of dissimilar. And we evaluated different encoder models and then applied the best performing one to encode a database of 1.74 million images. And then we wanted to perform laboratory experiments on this and see how does retrieval work in the lab. And this is where I came into the project. And so what we do is we encode our database as a DNA pool. We order that pool, and then we also encode Cas9 guide RNA with that same encoder model. And we introduce it to our DNA pool, and then through some molecular magic, we can selectively sequence things that were cut by the Cas9 molecule. And another important thing is that not everything that was cut we want to consider retrieved, because there's a lot of off-target activity and things are cut at different rates. And so we have to pick some threshold and say anything above this activity rate is what we consider to be retrieved. And then we can look that up and look at the files. So when we started to do these experiments, Jeff had a very important and foundational research question, which was, can we find Bigfoot in our database of 1.7 million images? So we used Bigfoot as a query, and then we also have a cat one for comparative reasons. And we actually performed these retrieval experiments. So kind of a complicated figure here, and there's a lot going on. But essentially what we did is we took all 1.7 million images in our database, and we binned them by Euclidean distance to our queries. And then we also binned them by how much Cas9 activity, either in a simulated setting or in the actual physical experimental setting, the query had on each of the sequence representations of the image. And then the colors here are basically just the proportion of images within a bin of Euclidean distances that fell into each of the different degrees of retrieval, we'll say. And so we have dotted lines here that represent our cutoff thresholds for similar, which is above the horizontal line, Euclidean distance, which is less than 75, and then dissimilar, which is below. And then we have retrieved, which is on the right, and not retrieved, which is on the left. So we have a table summarizing those categories here. And basically, a really good model would maximize all of the blue in these lines, or all of the blue rows, and then minimize the proportions in all of the white. And while our model does a great job at encoding so that we can facilitate retrieval of a lot or the majority of the similar images to the query, we also see a huge enrichment of dissimilar images, which is not great. So why might this be? Well, if we think about our encoder again, the result is that we reduced 1.7 million images into 457 encoded sequences, which is not good. The consequence of that is that a lot of images, if we think about them as bins, hold a lot, or a lot of sequences, if we think about them as bins, hold a lot of images. And so the bin with the smallest amount of images just had one, but then it just goes up from there, and the largest had 142,000. And so obviously those are going to have like all kinds of different feature vector distances to our query. And the result, of course, is that if we want to enrich for similar images, we're also going to enrich for a lot of dissimilar that are contained within that sequence that represents those images. And so what this figure shows is each line is actually a sequence, and it's ordered by its predicted activity to either the cat query, either the cat query or the Bigfoot query. And then each dot is an image that is represented by that sequence. So particularly with the Bigfoot, there's a lot of representation across a lot of different bins of similar images. And in order to retrieve some of them, like, we're just going to have to retrieve dissimilar, and that's not great. So, however, it still works pretty well. I mean, it's great to see that, like back here, for example, we really are like selecting for the most similar. And so we want to think about why this might be. The most likely scenario is that Cas9 has evolved to be hyperspecific, and that it's actually too specific of a molecule to use for similarity search, which at first might seem kind of counterintuitive, because if you think about something that has more off-target activity, you would think that would be the thing that would be retrieving a lot of dissimilar images. But you can really think about it in terms of, or like at the encoding level, to where if you have a hyperspecific molecule, it's going to reduce the amount of encoding space that you have to represent differences between images. So this graph just shows if you consider two sequences that are identical, as you increase the edit distance between them, compared to hybridization, you see a really sharp drop-off in the predicted activity between the two sequences, which translates to a really small encoding space. And so there's a lot of nuances in image similarity that you can't account for. And then just as a physical example, we can see if we have image A, and then we have image B that's similar to image A, and then we have image C that's similar to A, the encoder doesn't have a great way, if B and C are dissimilar, to represent those differences by sequences without also affecting A's ability to recognize or like retrieve both B and C within the database. So it's probably going to end up grouping them all together. Possible mediations to this problem involve using Cas9 as more of like a hierarchical thing, where we first separate the data set, and then we perform something that's more specific to subset the different retrieved pools of images. And then also using either a commercially available or engineered Cas9 that's less specific, and seeing what that would look like. But that remains future work. So with that, I want to thank all these people, particularly Nick, Lee, and Karen, who were the grad students who really did a ton of the engineering and architecting of this work. And yeah, that's all. So thanks.
All right. Thank you, Karina. Got time for one quick question. Gus.
Was that feature that you were initially using, was that like at the maximum size you could possibly encode DNA? Maybe not in DNA in general, but like, I guess for the purpose of your experiment, was that feature as big as you could have made it?
The feature vector, I think so. It was 4,096. Like the feature vector space was 4,096. But the Cas9 like space that you can encode those differences in is limited to only like 20 nucleotides. about. And so that's where the limitation comes in.
I see. Okay.
Yeah.
Thank you.
All right, thank you, Karina. Okay, up next we have Chandler Peterson, who's going to be talking about Boolean search using strand displacement in DNA data storage.
Yeah, so everyone, my name is Chandler Peterson. I am a graduate student in Georg Sig's group, and I'll be talking about Boolean search using strand displacement in DNA data storage. So as Karina mentioned, so DNA has emerged as this ideal storage medium due to its high data density, durability, and enduring relevance in the biomedical sciences. So briefly, data such as pictures or words, here represented in binary, are, are encoded in DNA and then synthesized. And then when data is retrieved, you can use PCR to amplify the pool, and the resulting DNA is sequenced and decoded. Of course, it's not practical to sequence your entire database if you only want a specific file. Here in the case of PCR random access, the specific primers are unique to the file and are targeted for amplification prior to sequencing. Now, random access is important, but oftentimes we need more sophisticated and intuitive ways to search for data in our database. Here, we utilize the promiscuity. So here, as Karina mentioned, you can utilize the promiscuity of DNA binding and do content-based retrieval based with hybridization. And another approach encodes data in nicks and spaces on a DNA register, and this method allows for in-memory computation with DNA strand displacement. Though this has a lot lower data density due to the encoding scheme. And then finally, there's a method using Boolean search using probe hybridization and different rounds of fluorescence sorting to perform queries. So here we introduce a new approach to integrating strand displacement computation with high-density DNA data storage, where both data and logic gates are encoded in compatible formats. So that last approach, using Boolean search, is a rather intuitive way to search a database. For example, let's say we have a database of haiku poems from Japanese poets, and we want to find a certain haiku based upon the file attributes. So here, each haiku has two attributes, a poet and a season. And maybe we want to select a certain one out based upon those two attributes, or a subset based upon a combination of attributes. So we will use DNA strand displacement to query our database to select file subsets based upon those attributes. So strand displacement compatible high-density data storage is going to consist of a couple different components. First, we have a strand displacement logic module that computes some sort of Boolean circuit. And then we need a compatible file format. So I will introduce this data gate design that consists of a file and an address sequence. And then finally, we want to read out both our circuits and our files with DNA sequencing. So now I mentioned DNA is a good medium for data storage, but it's also useful for implementing computation. In addition to being biocompatible, it's this like base pairing property of DNA that is particularly attractive and allows us to make, it's very predictable, and it allows us to make it an attractive engineering material for designing biochemical circuits. So when designing DNA constructs, it's useful to introduce the concept of sequence domains. Domains are groups of DNA bases that have some sort of functional behavior. Here, we introduce this partially double-stranded complex that we'll use to describe DNA strand displacement. And it has two functional domains, a total domain and a branch migration domain. And these domains are useful for strand displacement. And here, I will present these just in their domain form for simplification. So DNA strand displacement reactions are a commonly used and well-understood molecular primitive for DNA computing. In a basic strand displacement reaction, we have the template strand in green and the branch migration domains here in purple. There are three main steps in strand displacement. First, the total domain binds, and this initiates a branch migration process that kind of proceeds across the duplex, and this kind of is a random walk, and eventually the invader wins out and releases this output signal. So we can use this sort of input-output relationship to construct more sophisticated computational behaviors. one of those is we can create an AND gate. So in this AND gate, we have three DNA species, an input D0 and D1, and an output D3. And then we can create these strand displacement gates, which is just a series of these tool-mediated strand displacement steps that allow us to take an input D0 and D1 and result in an output D3. So the join gate joins two input signals, and the fork gate forks an output signal. And then we also have some helpers to help this reaction proceed forward, and an end cap domain that appends to the gate upon completion of the reaction. So to distinguish reacted versus unreacted gates, this end cap domain is crucial. You cannot tell after you sequence the gates if it's been reacted unless you have this end cap present, because it basically changes the state of the gate and allows us to see that it has been fully triggered versus non-triggered gates. So here you can see in the biophysical assay that a reacted gate is lengthened by 20 base pairs, and this causes an upshifted band that shows that our gate has been fully triggered. And this is also visible in DNA sequencing. So to make this state change permanent, we ligate the gates prior to sequencing, add some additional sequencing adapters, and then we can read out all of our gates in parallel using DNA sequencing. So to show the function of an AND gate, here we have a gate with fluorescence readout on the left and a sequencing readout on the right. Here, the fluorescent signal corresponds to the amount of output present in the system. So the high signal indicates that the gate has been triggered, and similarly for the sequencing readout, the number of reads present after sequencing indicates like the number of species of a triggered gate. So here we can see that both the fluorescence and the sequencing readout correlate nicely. And you can see that only in the case when both inputs are present do we get full gate activation. So now that we have this one unit that works well, we tried 6 parallel AND gates, each sharing inputs and sharing outputs, such that we can trigger subsets of different gates with a limited number of inputs. So you can see in that last condition where all 3 inputs are present, we get full activation of all 6 AND gates here in this heat map, with these dark squares representing maximum number of recounts for each condition. We can also look at each gate individually and see that they all operate with correct AND logic. So only in the case when both inputs are present do we get this full activation. So each gate in this pool is reacting as we would expect. So now that we have this reliable way to perform Boolean circuit computation with strand displacement, and we have this entire subfield of work that has made high-density data storage in DNA feasible, outlined by Karina, the question becomes, how can we leverage strand displacement for random access and do this sort of Boolean search problem where we're using our inputs as file attributes and reading out some data? So a very similar circuit to the one we just looked at might look like this. We have DNA inputs that are our file attributes. And so if you have Autumn and Bashō, this will map to a distinct output that is an address that will then interface with the data module. And that will lead to our data. So this data gate looks like this. We have a file fragment on the right and an address on the left. And each data gate consists of multiple file fragments. And this is just to make the gate shorter, such that a single file can have any number of file fragments, but they all have the same file address. And you can do this for any number of files in your database. So say that we want to select file number 2, just directly with the address strand, we would add in the address for A2. This would make a strand displacement strand displacement step that would allow this end cap to come in and append to the gate, showing that it's been triggered, and then we could proceed with ligation and sequencing to read out that gate. So what we did just to test that our gates were working experimentally was that we targeted each interval of files with just the address strand to see if we can just read out those gates to make sure our data gates were working appropriately. And we can see that in each case here, along the left, if the correct file address was input, then the correct interval of data gates would activate for each query. So in the case, yeah, Bashō, all the different seasons activate, and only those intervals light up with a very limited background. And this also works if you want to do multiple at once. So before composing our logic module with our data gates, we check to see that the programmable logic array is working as expected. And this is largely the case. So now that we see that our strand displacement-based logic is working and our data gates are behaving correctly, we can compose the two and read out some queries. So here's an instance of random access using Boolean search where our query is Bashō and autumn or Busan and autumn. And we can see that, again, only the interval for Bashō and Busan over autumn activates with a relatively low background in comparison. And now we want to see does this work for every combination in our database. So we do a very similar experiment, and just to see that all these intervals light up, and we can do this essentially the same experiment that we did when we targeted them just using the address, but we can instead use the file attributes. So we are actually accessing each of these file intervals using the attribute inputs rather than just the address strand. So now we want to do maybe some subset selection and do some algorithmic querying based upon subsets of file attributes that we add. So one interesting one is all seasons and Bashō, and you can see in this case we get activation of every data gate fragment corresponding to every single season in the interval and just the poet Bashō. And similarly, autumn and all poets, where we get activation in both of the poet sections, but under just the autumn intervals. So future directions for this project include using array-synthesized DNA, which is in a DNA that we can produce in mass for relatively cheap, and this will be useful in scaling up to larger circuits in databases where we can perform more complex queries over larger databases. And then we also want to make sure that, and I didn't address in this presentation, that we can actually read out these files and reconstruct the original message correctly. So we'll perform PCR random access and decoding, and also extensions to isothermal PCR or PCR-free random access, where our sequencing adapters are actually encoded in the appending end caps and flanking regions. So we only need to dump the reaction into the sequencer, and only reacted gates will get sequenced. Thank you. And I want to acknowledge the CLIG lab, people in MISL, Gwen Root and Chris Takahashi for helping with the encoding work, and then my advisor Gary CLIG, and of course members in the Thatchuk lab. Thank you.
All right, thank you, Chandler. Time for a quick question. No, not so fast, Chandler. Any questions? Up there.
It seems like as doing a query causes an actual physical reaction. Is there an upper bounds on how many queries you can run on a database before something gets permanently changed and broken?
Potentially, yeah. So you can see there's a decent amount of background here, and that's potentially compounded by the number of DNA input species that are present in the system at a time. So you might get more background in certain queries than others. So as we scale up these systems, that's something we're hoping to explore is how robust are they to this leak reaction when the computation, the circuits themselves and the queries get more complex.
All right, thank you, Chandler. Okay. Up next, we have Katie Pierce that will be talking about building fast, robust molecular circuits and amplifiers.
All right. Hi, I'm Katie. So why compute with DNA? I think you've already heard this a little bit, but essentially we would like to build logic systems that can interface with biological systems, and DNA is the information molecule in biology, and we'd also like to build diagnostics. For example, I've heard a lot about COVID tests, maybe that's old at this point, but things like that based off DNA sequence. So DNA circuits already exist. This is one example from some previous work. So we can build logic gates with DNA strands, and these can scale up into larger circuits where we string these logic gates together. So let's look at just one AND logic gate. It's off with no inputs, off with either input, but with both inputs, it turns on as expected. So one thing you might notice is this is a pretty long time scale. That's in hours. So we would like to speed this up. One way you could think about speeding up a chemical reaction is adding a catalyst. So we found this catalyst, and it turns out it does in fact speed up this reaction, which is exciting. The only issue that you might notice here is these off cases now also get sped up, and what used to be maybe a slight amount of leak that was unnoticeable on the time scale is now a very large amount of leak that is also sped up. So this is a problem if you're thinking about building a larger circuit, because just in one of these gates, you're going to have this large amount of leak that can carry through the whole circuit and compound until you can't distinguish between signal and noise. So how do we fix this? Let's look at just a single translator gate from X to Y. So you already heard from Chandler about DNA strand displacement. So I'll try not to spend too long on this, but essentially you have an incoming signal strand that comes in, and the whole complex goes up in energy. That's an energy diagram on the left. And then your energy sort of goes up and down till eventually you release the output strand and you go down in energy, and that drives the reaction forward. So here is a video of that. And essentially the same thing is happening, and that red part there is new domains or new bonds that are formed that drives the reaction forward. So let's look back at our translator gate. Just a summary is that eventually we want to get to a lower energy state, but we have to overcome this energy barrier, which our catalyst is going to help with eventually. So the leak we were talking about in our AND gate happens when if you have some downstream reporter complex that you need to eventually read out your signal. In the absence of any input, there's a reaction that can happen where essentially you separate your reporter complex and you get fluorescent leak. So how do we make this less leaky? This is work that I worked on and presented with Tiernan Kennedy, who's another grad student in the lab. But it turns out if you add these A and B intermediates, you can prevent leak. So instead of going directly from X to Y, we go to A and then to B and then eventually to Y. And this introduces a form of kinetic proofreading. So does it work? In the lab, yes, our off case stays off and our on case turns on. So we can apply this to build a leakless amplifier. So instead of going from X to Y, we're going from X to X. And we make it robust by adding these A and B steps, and in each amplification cycle, we are releasing fluorescent signal. So as you keep going around this amplification cycle, you build up more and more signal until you can achieve detection. So we can use this to detect a very low concentration signal in just 10 minutes. And this is work I did with Zoe, who is also in our lab. So essentially, all you need is the catalyst, the amplifier, and in 10 minutes you have detection, which has maybe some implications for diagnostics later down the line. Let's go back to the AND gate. So with the catalyst, we said that the leak was unacceptable. But using these same techniques I've shown with the translators, we were able to build a more robust AND gate where we could use our catalyst and the leak cases stayed off, but we get our speed up. So this time course is now much faster. So we are hoping to use this in FPGAs, combining our AND gate with our translator to make these larger circuits. And then we can introduce this into an automation system where you essentially program on the computer, then you mix things in a liquid handler, and then you're able to read out in a plate reader. And we will also be testing this out in Chris's class that he is teaching next term, that I will be TAing, and seeing if people can program these simple gates or simple to program gates. So we've talked mostly about small components. So when we scale this up, how do we make our circuits? So we need the fridge compiler, which Lancelot, another grad student in the lab, has been working on. So essentially you have a bunch of DNA and a bunch of gates in the fridge, and you want to implement a function. You take the fridge compiler and it will give you the smallest inventory you need so that you save on DNA to implement your function, and then you can mix it up in the lab and get your output. So thank you to my advisor, Chris Thachuk, people in our group, Zoe, Tiernan, and Lancelot, who I've been working with and whose work was also in here, our other collaborator, Paul, and then everyone in MISL.
All right, great. Thank you, Katie. Time for a quick question as well, I think, if any questions from the audience.
How much, I missed the x-axis, how much kinetic delay does the intermediate gates add, even with compared to the very leaky version and the now non-leaky version with the intermediate delay?
That's a great question, and I didn't really explicitly show this. So here, this isn't really a good comparison to anything. I didn't show the comparison case because that's just very leaky over this time scale. The AND gate that we ended up building was actually significantly different in architecture, so it's not a very fair comparison. And I think you also saw some of Chandler's gates, which were an even different architecture, and it all depends on the concentration. But you do get slight slowdown when you make it more robust. And in the translator, it's easier to say it's a slowdown of 3x because you're adding, or you're turning one step into 3. Here, it's a bit more tricky. But we hope to, with the catalyst, we're hoping to get speed up of anywhere from 10x to 100x, depending on the system.
I mean, it's switching on there in half an hour versus
Right.
20
Yeah.
So
It's a different architecture, so it's not necessarily a fair comparison in this figure.
All right, thank you, Katie. Next up, we have Jason Hoffman, going to be talking about the fluorophone.
Yeah, thanks everyone. I'm a fifth-year PhD student, kind of across both the MISL lab and the UBComp lab, and this project is a collaboration across both. It's kind of in a little bit of contrast to the earlier projects, which are core molecular computing technology. This is more about, can we bring this technology to a wider audience and be more available to be used more broadly. It was also presented as a poster at DNA 29 this year, by my friend Katie Pierce, who you just heard from. And so I wanted to start off by talking a little bit about what inspired us to start down this path. If you didn't know, I spent a couple years early in my PhD during the pandemic trying to see if we could track the spread of viruses, specifically COVID-19, using the infrastructure of metro buses in the Seattle area. And what we did was we went in and grabbed the filters from the buses that are pretty much always running to clear the air and make it healthier to breathe in those buses, and then brought them back to lab to run a PCR test somewhat similar to what you would do with your own personal sample. And we were trying to see if we could build a map of spread of the virus within the city, or potentially create an early viral monitoring or surveillance system similar to wastewater sampling. And we developed a method of collecting and extracting. However, this method was somewhat cost prohibitive in actually answering the research questions we set out to answer because of the amount of time it took in lab. So while we could go and collect a sample, go to the bus depot for an hour, grab the sample, it then took a couple days and multiple grad students, 12 hours total in lab just to process a few of those buses with the current PCR methods. So that led to the question of how can we improve this for future studies in environmental detection? And if we can solve it in a useful way, can it generalize to other applications like medical diagnostics, field research, and other things? And so, in order to start thinking about this, we looked at what are the problems that prevent us from doing this. One is molecules are small. So as you saw in a lot of the DNA strand displacement work that people did and molecular computing, there's always some way to transduce that signal from something that we can't see to something that we can sense and or read out. And typically we have some sort of hardware or a colorimetric, like visible output that can be seen with the naked eye. So a lot of people have mentioned fluorescence so far today. What that really means is you have a reaction that's set up so that certain molecular reactions happen, and if they happen in aggregate with a lot of molecules, this particular attachment to, this is a DNA strand displacement reaction, starts to glow, and an aggregation of those can create something that can be seen by some instrumentation. A similar thing happens with colorimetric, except that output is visible to the naked eye without a sensor, and I'll talk a little bit about those sensors in a second. So yeah, so what are those things? In lab, we typically have some specifically constructed devices that can sense these fluorometric outputs, and they're very general purpose for a lot of different lab tasks. However, you don't really see them outside of the lab. This is not something that I can buy it for several $1,000s and set up my own lab, but people don't in general. And so there's things like qPCR meters, which can run a PCR reaction and measure the change over time. There's things like plate readers and other devices. One that I'd like to call out, which you will hear about in Daphne's talk, is called a nanopore sensor, and those things have made great strides in becoming more accessible and lower cost. Still, it costs $1,000 or so to get set up, and a lot of money to run that test as well. So we're moving towards it, but what could be even more widely applicable? Coming from the UBComp lab, of course, I thought of smartphones. Can we build a smartphone detection method where you can leverage the sensors that are already built into the phone, namely the camera and the flash, maybe other sensors as well, and use those to kind of replace some of the functions that we use in lab work. And so the idea would be any chemical reaction, right now we're focusing on a few specific ones, but any that do fluorescence or colorimetric output, can we sense those with a simple smartphone setup? Yeah, and so as we were looking into this a little bit, there are a lot of solutions out there in research, and as I've been reading more about it, we've been getting inspiration, and also realizing that every single one so far that we've encountered requires some sort of hardware or some sort of setup or thing that's like not quite as simple as it might look. And there's good reasons for this, but it's just interesting that there's still some space to move towards even more availability and ubiquity. In addition, one of the things we do, at least in the HCI side of research, is test for usability, and there's not any, as far as I can tell, usability tests on these types of systems. They're kind of assumed. And then, so we started setting up our own system. We started off with this fluorescence readout, which is the most common readout used in DNA computing, at least, and a lot of DNA diagnostics and things. And so we did realize at this moment we haven't been able to remove all of the attachments, but we built kind of a minimal attachment, which is these little goggles that you kind of slide onto your phone, and they're 3D printed. And you can swap in different filters, and the filters really help with the mechanism of a fluorophore, which requires you to send in some energy of a certain wavelength and then read out a different wavelength, which is known as a Stokes shift, to actually see the fluorophores light up or glow. And so, Zoe also helped with a lot of this research. And we also started using these paper-based tags in lab, something that postdoc Jiaming Li had developed in lab. And as you can see, when we put the goggles over the phone, kind of controlled for light by turning the lights off in the room, we were able to see that characteristic glow of the fluorophores just kind of through the smartphone lens. You can't see this with your eye because your eye picks up all of the wavelengths, but when you use the phone filter and just put this on the phone, you can see that this particular experiment, we used different amounts of time to have the molecules interact and produce more fluorophore over time. And so you can see, especially in the green wavelengths, that the phone picks up the signal in about 15 seconds or so. But we wanted to take this a step forward, of course, and Katie was doing some awesome work with colorimetry along with Zoe as well. And so we wanted to see what we could do with zero attachments, so just the phone itself. And for this, we picked up a reaction that you can actually sort of see with the naked eye. But we wanted to see if the phone maybe is perhaps more sensitive or can pick up things, especially if we tune the phone's camera sensor with an app. Can it pick up something more sensitive? We were able to go down to about 2 nanomolar concentration so far, and we're still working in lab to kind of optimize this and potentially do it on a paper-based method as well. But really all it requires is the phone with the app running and then a cardboard box to control for light, which is pretty nice. And in order to do this, we've also started developing some processing algorithms. So we start with an example of the tag. When it is placed with some fluorescent output, then you put the filters on, you take a picture. Once you do that, you process it to find the locations and then see what the color change is. And so that's where you see the most bright spot is the furthest along in terms of time, and the second most bright spot was about half that time. We're thinking about different ways to do this in a more, I guess, sensitive manner, but right now we're just doing color change, considering things like machine learning, concentrating in a certain locality of the ticket, and lots of options right now. And that's where we get to this. This is a research project in process right now, in progress. And so we have lots of ideas around using the camera or using other parts of the phone, such as the smartphone touchscreen. How do we apply this to diagnostics? Do we, use the front camera versus the back camera? There's lots of options here, and I wanted to also share this with you today to see if there's anyone who has other ideas that they, might think this might be useful for. And yeah, feel free to reach out, especially if you have like an application that you're like, hey, I want to run this diagnostic, and I think it'd be really cool to try out. Ears are open. And I also wanted to share that a lot of people have contributed to this work, and it was really awesome to work with them. So thanks to everyone in MISL Lab and UBComp Lab who've helped out. So yeah.
All right. Thank you, Jason. Time for a quick question. Anybody? All right, I had a quick question. The cost of the what you are using with, as far as that filter setup that's 3D printed and whatnot, what is the cost?
So yeah, a 3D printer setup is kind of complex to begin with.
Or just like the piece with the filter, like
the filter itself. Yeah. Those filters were about $20 each, so a short pass and a long pass filter so far. So it is still more expensive than I'd like it to be, but we have some ideas. I guess I didn't mention, there is inbuilt filters in the camera. If you're familiar with how a camera works, there's these things called Bayer filters. So we're considering trying to use that to do the filtration without the filter.
Well, it's a lot cheaper than a plate reader, so that's good.
Yeah.
Cool. All right, thank you, Jason.
Yeah.
All right, and the last speaker we have up is Daphne Cantos Georgios Heights. Your mic is right there. And she'll be talking about modeling and decoding nanopore signals for single molecule protein sequencing and barcoding.
So long title, but breaking it down, we're trying to work towards this big goal of protein sequencing. So proteins are chains of amino acids that are then folded up into these big, complicated 3D structures. And protein sequencing is the task of, if I have a physical sample of a protein in a test tube, can I then linearize it and then figure out what the sequence of amino acids are that made up that protein? So you probably heard DNA sequencing. They all used it a lot in their, I guess except Jason, in their talks. It's very important, helps a lot of biology research, but we're nowhere near as good at it with as protein sequencing as we are with DNA sequencing. So the way that one would do protein sequencing now typically is with mass spectrometry. And mass spectrometers are these really big, really expensive, really bulky devices that require you to have a lot of your sample in order to analyze it. So they can't do a single molecule analysis. Also, we just generally hate them in our lab. We like nanopore devices, which are these next generation DNA sequencing and RNA sequencing devices that we're trying to hack to instead sequence proteins. So these are commercially available devices. that even Karina uses in her research for DNA sequencing. And the way that these work is you have a membrane with a small hole in it that we call a pore or nanopore, and you apply a voltage to this membrane, and ions are always flowing through this pore. And at some point, the biomolecule that you're trying to analyze will come along and a motor will pull it through the pore. So when that molecule is going through the pore, that blocks the flow of the ions. And so as those ions are their flow is being blocked, that affects the current. And so you have a really small ammeter across this membrane that's reading the current over time. So you can look at this current trace and then get information about that molecule that was going through. And you get information about like the size and this charge at each point in time. So then the data that we get from the device looks something like this. So then we can then look at this data and then try to make sense of it. So at the beginning, we get this really high current that corresponds to where there's like no resistance and ions are just flowing through. At some point, the molecule will come along and go in the pore, and that's when the current drops down. And at some other point, the motor will come along and pull the molecule back out of the pore, and that's this little bit right here, but it's relatively slow when it gets pulled back up, and that's what we're interested in. So if we zoom in, we can see something that looks like this, and this is a protein going through the pore, and this is the part that we look at. So one of the reasons why analyzing these sequences, or these signals is complicated is because this motor is a stochastic thing. It's not like a mechanical motor that takes one step at uniform times. So all these traces up here are the same protein going through the pore in the same experimental conditions. And you can see it's generally the same thing, like monotonic increasing, then a little dip down. But the time scale on all of these is different, and the time scale on each step is different. So, for example, this one had really low or really short time at like this step, but then really long at this step, whereas other ones were really long at that sequence. So that means that in order to analyze these sequences or these signals, we have to do dynamic time warping, which is just kind of a typical dynamic programming algorithm to align these signals to each other. The other reason why analyzing these signals is really complicated is because amino acids are smaller than the pore. They're even smaller than nucleic acids, which these pores were optimized for. So roughly 20, maybe more, maybe less, amino acids fit in the pore at a single time. So that means a bunch of amino acids are affecting the current. So in this plot, we have 4 different proteins. One's purple, one's blue, one's orange, and one's red. The different traces of it aligned to each other. And so G is a really small amino acid, Y is a really big amino acid. And so these proteins all have the same sequence, except like the blue one has these mutations, the purple one has these Ys here, and such. So if you look at this sequence, for example, where you have a few Ys at the beginning, the red one before, you can see that its current is low at the beginning because it has those big bulky Ys, but it's also not just low here, it continues to be lower than these purple and blue ones for a while, and that's because of that window effect. So analyzing these signals is complicated, and we design these very specific synthetic proteins to be able to begin to characterize what the effect of each amino acid is. So the way we design these proteins was it would have repeating units, and in between each unit, we would change one amino acid at a time. And on either side of this unit, we would have 2 tyrosines, which are 2 Ys, which are these big bulky amino acids that would drop the current down a lot. And so those drops act as landmarks, and then we can look at these areas in between the YY dips where just one amino acid has been changed at a time. So these little areas where one amino acid has been changed, we call variable regions. So we can see here the difference between like this blue bit and this yellow bit is because of the difference that a D has versus like H has. And we made a bunch of these proteins and sequenced them so you can see each amino acid and each amino acid in 2 different contexts. And then once we got this data, we could then start to make a really simple biophysical model predicting for a given sequence, what would this signal look like? This looks complicated, but I'll break it down. So we would look at a roughly 20 amino acid window and create a vector that would be a linear combination of the volume and the charge of each amino acid, and create a vector of like each amino acid in that window, and then compute the dot product of that vector with a parabolic weight so that the amino acids at the center had the most weight. And then from that number, we would get a value, and we plot that value for each of the windows of the protein. And this very simple model does a really good job of modeling our synthetic proteins. So here's an example of a real signal in black that's been filtered to the model signal for that sequence after they've been lined together. So this works well, and that was cool. And then the other type of modeling we've been doing was we made a machine learning model to predict for a given squiggle, to predict what was a single amino acid that's been changed. So doing some proto-amino acid sequencing. So the way we would do that is we would take the raw signal, filter it, downsample it, and then collect these, or extract these variable regions and featureize them, and then plug it into the machine learning model of choice. And then see what our accuracy was at assessing like what amino acid that was changed in that part of the protein. And so these numbers are pretty good, especially if you compare it to other times that people have tried to do amino acid characterization with nanopores. So if you're just trying to distinguish a few amino acids, it's really easy. If you're just trying to guess what amino acid was out of all 20, it's not so easy. There's a few reasons why it's not so easy. One of them is that a lot of these amino acids look very similar to each other. For example, leucine and isoleucine are just isomers of each other. The other thing is that ground truth is changing on us because the proteins can be chemically modified between the time that we synthesize them and the time that we sequence them, which also complicates things. Also, I mean, it's just difficult. So like our 20-way classification accuracy was around 30%. But the nice thing is that we found a sequence that causes the motor to lose its grip and the protein falls through the pore. And when it falls through the pore, everything is pulled back out again, and we get rereads, and those rereads increase our accuracy. So we had this like EPP sequence that we repeat a bunch of times, and this purling causes the motor to lose grip, and then we reread it. And then so this is a simulation of how the rereads would improve accuracy. So like with more rereads, accuracy goes up, and that was exciting. So all this modeling is just with synthetic proteins. It's not with real proteins. Though we're still interested in working on real proteins, we were thinking about potential applications that we can use for the fact that we can sequence these synthetic proteins. And one of them is to use these synthetic proteins as barcodes. So if we can very reliably and accurately identify these synthetic proteins, we can add these little proteins onto other things and then be able to identify those other things. Those other things could be proteins of interest in like synthetic biology experiments, or one other use case that we're starting to use already is in our experiments is to increase the throughput of our experiments so that we can test multiple proteins at a time in one experiment instead of just one protein in an experiment, because in these devices we have a bunch of different pores. So yeah, hopefully working towards reading native proteins too, but even just the fact that we were able to read these really long synthetic proteins accurately was really exciting. And so with that, I'd like to thank all the other people who've been working on this. They've been really awesome. And yeah.
All right, thank you, Daphne. Super exciting work. Any questions? Got time for one question here. Let's see. Gus.
Sorry if this is a basic question, but is it, so are nanopore sequencers built for both proteins and for DNA, like alike?
No. So they're designed for DNA and RNA. We've just been hacking them to instead work with proteins.
I see. So did you actually have to do something physical to the nanopore sequencer?
So we didn't modify the sequencer, but what we had to do was add this protein that pulls it out. And then, I don't know, there's probably, Mateus and Jeff could tell you if there's other experimental changes.
It's not for DNA.
Yeah, there is a motor that you have to add to DNA, but we use a completely different motor.
I see.
Yeah. And then we also had to add some like extra tags into our proteins for things to work and stuff like that. Yeah.
Cool. All right, thank you, Daphne, and let's thank all the speakers again. That was a great session. And yeah, that concludes the colloquium. Thanks, everyone.